The second part of Chapter 11 then deals with several different related topics or issues that are all tied together with this idea of gene expression and gene regulation. The first is signal transduction. Signal transduction is how signals are passed from the outside of a cell to the inside. And so basically, it's the way that cells chemically monitor their environment. And we know, based on our last topic, that the environment may influence gene expression. And so the signal transduction is going to play a role in genes being turned on and off as cells chemically monitor their external environment. Also, cells being cloned or organisms being cloned has to do with this idea of gene expression and sort of resetting a genes, turning genes on and off. So we'll take a quick look at some cloning issues, and then finally we'll finish up with cancer. We've talked about cancer already as being that disease due to mutations and problems with the control of the cell cycle, that a cancerous cell is a cell that grows out of control. Well, the genes associated with causing cancer are genes that then are going to be undergoing differential gene expression, or we're going to change the expression of genes that may lead to a cancerous cell. So we'll look at these three main topics: signal transduction, cloning, and cancer, all related to gene expression. So the first of these issues is signal transduction. And signal transduction really has to do with how cells monitor their external chemical environment. And if I'm looking at your body from the perspective of the cell, the external environment of that cell are your bodily fluids, the chemicals around that cell. And when certain chemicals are present, they will bind to the cell and signal to the cell that the environment has changed externally. And so, for example, let's look at insulin. Insulin is a hormone produced by cells in your pancreas. And so as that insulin is produced, it circulates in your blood. Certain cells then have insulin receptors, a receptor protein on the surface that will only bind to insulin. And so if insulin is present, that insulin will bind to that receptor protein, and that will cause the signal transduction pathway to be activated, which means basically that receptor protein will undergo a chemical reaction, which will cause another chemical reaction, and so on. And so this whole series of chemical reactions will occur, passing that signal of insulin being present from the outside of the cell, chemically passing that signal to the inside of the cell. And once that happens, then transcription factors are going to be produced. And remember, transcription factors help to increase or activate genes or gene expression. And so normally certain genes are turned off, but if the signal pathway is turned on, if it's activated, these transcription factors are going to be produced, which are going to cause then certain genes to be activated, and the cell is going to produce new proteins and change its metabolic pathways. And so, for example, with insulin, when insulin binds to these insulin receptors, this insulin pathway of signal transduction begins, and cells then will start producing proteins that allow them to absorb sugar out of your blood. And so the presence of insulin then is going to result in your blood sugar being lowered in this particular case. And if we just look at a figure kind of depicting this, again, our receptor proteins are on the surface of the cell. You have hundreds of different receptor proteins that are specific for only one type of chemical. And so insulin receptors are going to bind that insulin, cause a whole series of these relay proteins to be built through this whole, again, signal transduction pathway of chemical reactions. The final stage is a transcription factor is produced, which, remember, are going to bind to DNA and activate a gene. So turning on gene expression, so that DNA then will get converted into RNA, into protein, and so we've got a new protein being expressed due to our signal transduction pathway. And all cell types exhibit some form of this signal transduction pathway, so we know it's a very ancient form of the way cells communicate with each other by these protein receptors and these various signal chemicals that may bind to them. And your book talks about the interesting example of yeast and their whole mating process. And yeast are single-celled fungi, and their whole mating process is controlled by these specific signal transduction pathways. Now, there aren't male and female yeast, but instead we call them yeast types A and types alpha. So chemically, there's kind of two different forms of yeast. And their mating is going to be controlled directly by these pathways of signal transduction. And what happens is type A has alpha receptors on its surface. Alpha has type A receptors on it. And so if type A yeast is present, it will release its type A protein out to the environment. If there's an alpha yeast nearby, it has type A receptors on it. The A binds to it and undergoes one of these signal transduction pathways to activate genes in that type alpha that will cause that alpha type then to start growing toward the type A yeast and eventually fusing together and mating with it. And then just the opposite occurs for the type A. It has alpha receptors. If an alpha yeast is nearby, it will bind with the alpha proteins and cause it to grow toward it. Okay? And we know that many of our eukaryotic cells then and multicellular organisms have taken this basic unicellular process and modified it. And we see this very commonly, for example, in both multicellular and unicellular organisms, our signal transduction pathways. And here's just a visual way to see our cell signaling in yeast and this idea of our mating. Again, the alpha has type A receptors on it. If A is being produced by an A yeast, it binds to that alpha receptor. The opposite would occur in the type A yeast. It's going to bind its alpha proteins on it. Each of those proteins then, once they're bound, will cause a signal to be passed on, transcription factors released inside the cell, causing those cells to grow toward each other and eventually fuse together and to mate. Our second main topic today then is the idea of cloning and cell specialization. And we talked a little bit last time that different cell types have different genes turned on and different genes turned off. And so all of our somatic cells are all genetically exactly the same, but it's gene expression that makes each cell type different. And so we know then that cells that are differentiated or specialized then tend to stay specialized. For example, in animals, if you've got a skin cell, every time it undergoes mitosis, it'll make more skin cells. It can't make more liver or nerve cells. It's only going to make skin cells. That's not the case necessarily, though, for other organisms. And so we know that even though certain genes may be turned on or off, again, all of these different cell types are basically clones of one another. And this can be taken advantage, in fact, in plant cloning. For example, we can take a root cell of a plant, grow it in a petri dish. If we treat it with the right hormones, those cells then will grow into new roots and stems and leaves and grow an entire new plant just from that single cell. And that shows us that that root cell has all the genes necessary for every cell type in the plant. In certain animals, we see this with animal regeneration of limbs. So, for example, if a starfish loses an arm, it will grow a new arm. And so those cells that are present will grow and differentiate and replace those that were missing here. And so all of these involve what's called de-differentiation, which means these specialized cells can unconvert or basically turn their genes all back on and reconvert back into a stem cell, that basic cell type, and then re-specialize into new cell types. And again, if we think of our plant example, that root cell can unspecialize into a basic cell type, and then different types of different cells on our petri dish will re-specialize into roots and stems and leaves and so on. Okay? Now this doesn't work with higher vertebrate animals like ourselves. In our cells, this doesn't unspecialize. We can't take a single skin cell, for example, and grow a whole new you by growing it up in a petri dish. If we do that, we'll grow lots of new skin cells, but we won't grow new nerve and muscle and so on. And so some organisms cells can unspecialize and respecialize, like plants and some of the invertebrate animals. And this is just looking at that plant cloning example. And again, people in the botanical industry and horticulture industry have been using this for years. When you go to Home Depot or you go to a nursery and they've got 100 azaleas and all those azaleas look exactly the same, the reason is they're all clones of each other. Okay? If you go to the florist and you buy a dozen roses and all those roses look exactly the same, that's not an accident. Those are all cloned from one another. And what happens is if I got, let's say, a rose that has a particular shape and color to its flower or fluorescence, I want to patent that, and then I'm going to take that rose and I'm going to clone 1 million copies of it, and then I can sell those 1 million copies worldwide and keep cloning it and have that specific type. And because they're clones, I know all the plants are going to produce the exact same flower with this exact same colors and so forth and shape. And so to do that, you take a cell, you grow it up in a petri dish, and it will unspecialize. So again, we're going to affect its gene expression. All of its genes will now be activated, and then by treating that clump of cells on our petri dish with different hormones, some of them will become stems, roots, or leaves, and we can grow a whole new adult plant here. And so again, our gene expression is certainly changing depending on different hormones we treat the plant with. And while with vertebrate animals, our cells can't do that. Again, our cells cannot unspecialize and respecialize. We certainly can clone vertebrate animals by certain biotechnology processes, in particular one that's called somatic cell nuclear transfer. And in somatic cell nuclear transfer, the name kind of describes what's going to happen. we're going to take an egg cell and remove the nucleus. Okay, so we end up with basically an egg cell with nothing but cytoplasm inside, and then we're going to transfer the nucleus from a somatic cell, and usually a skin cell is used because skin cells are just easy to get, and we're going to implant that nucleus inside the egg cell. And something about the chemical nature of the cytoplasm of that egg cell will reset or reactivate all of the genes in that somatic cell nucleus. So now I've got a cell that's basically a stem cell. It's unspecialized. Then if we grow that up into an early embryo, and usually we're going to grow it up into maybe a 16-cell stage or 32-cell stage, and we can collect those cells. Once we have that embryo, there's kind of two options. It could be that we're trying to produce a clone. We're trying to produce a whole other organism, and that's called reproductive cloning. Okay? I would maybe take that embryo and implant it into a surrogate mother's uterus, allow it to develop, and then when that organism is born, it would be genetically the same as the original parent that we took that somatic cell from. And so that's our idea of somatic cell nuclear transfer. That's what was used to clone Dolly way back in 1994, and it's been used on lots of other organisms since, from livestock like cattle and pigs to things like pets like cats and dogs. Other individuals, when they're going to do this somatic cell nuclear transfer, the goal is not to clone the individual, but the goal is to get some stem cells, some basic cell types for some sort of medical purpose. And that's referred to as therapeutic cloning. So in therapeutic cloning, we're going to allow that embryo to grow up to, again, maybe the 16 or 32 cell stage, and we're going to harvest stem cells from that developing embryo, and then we can maybe take those stem cells and cause them to become different cell types, eventually, again, to treat some sort of disease. So here's just our process of somatic cell nuclear transfer. Again, it's a pretty old process, but people are kind of refining it and getting better at it. Basically, we're going to remove the nucleus by using just a little small pipette and sucking out the nucleus of that egg cell. We're going to insert our nucleus of our somatic cell. That somatic cell nucleus then is going to be reset. All of its genes are going to be active, so then it will start dividing by mitosis, and we can either use that embryo for reproductive cloning by implanting it and cloning that original parent, or we can grow it in a petri dish and collect a few stem cells and use those stem cells for some sort of medical treatment. So here we're looking at a video of the somatic cell nuclear transfer, and here you can see, this is an egg cell under the microscope, and the technician here has his pipette and he's sucking out the nucleus, and we can see the nucleus right there enter the pipette. And so this is our egg then with no nucleus, and so it's a denucleated egg with basically lots of cytoplasm and organelles. Now, as you might guess, there's a real art to this. Most of the time when a hole is poked in an egg cell, it's not going to survive, and so there's certainly a lot of technical prowess involved with making this work. Now here we can see the somatic cells being sucked up into another pipette. So this is the cell that we're going to clone. It contains that nucleus of a skin cell in particular. It's what they're cloning here. And this is an actual human clone. This is the first documented cloning of a human, and this is from about 2003 or so. And so here's our egg that's missing the nucleus, and so now we're ready to insert our somatic cell nucleus in it. And again, something about the chemistry of the egg resets our DNA in that somatic cell and reactivates all those genes that had been turned off in that particular skin cell. So now we can see here he's going to bring in his pipette, and you can actually watch the pipette as he pushes in here is going to kind of stretch the plasma membrane out and eventually kind of push its way into the cytoplasm. So here we see that stretching of that membrane. Once he gets that pipette into the egg cell, as we can see here, there it kind of finally punctures into the egg cell. Now he's going to inject that somatic cell nucleus into the egg. And here we can see he's starting to inject. Don't see the nucleus yet. Now here comes the nucleus now. The nucleus is just now right at the tip. There it is, being pushed into that egg cell. And again, that nucleus then, the gene expression will completely change of that nucleus. It will no longer make just skin proteins, but all of its proteins will be activated. What this lab then did with these eggs is they put them in a petri dish at the right temperature with some certain nutrients in it, and they grew them up till about the 8-cell stage. Now this group was not trying to clone humans and to get a whole bunch of new humans running around that are exact clones, but basically they were doing therapeutic cloning. And so again, this is the first documented case of a human being cloned, a human cell being cloned at least, and this was from about 2003 or so. Once they grew to the 8-cell stage, then they collected some stem cells and started doing some of their stem cell research here. Despite the fact that we're cloning these organisms, again, oftentimes mammals, other vertebrates like ourselves, that doesn't mean the clones are always going to be identical. Remember that our phenotype, all of our characteristics, are a combination of our genes plus some environmental influences and some issues dealing with development as embryos and cells develop. And so just because we have a clone doesn't mean it's going to be exactly the same as the original parent. And so, for example, if we look at this idea of reproductive cloning to produce organisms that have a trait that we like, whether it's agricultural, for example, producing livestock with a particular trait. For example, today we're seeing people look at trying to generate various types of cattle that produce leaner meat with less fat. And once we get that, then they're trying to clone those so that we can consistently get a low-fat meat product. It could be for therapeutic agents for animals, for example, that are producing certain chemicals that can be used in drug treatments. It could be for endangered species. If we've got species that are really rare, we may try to clone them to get more of them and then eventually try to get those different clones to survive and to mate with one another. Or sometimes we may even see pets. And this is an example of the first cloned cat. The mother there is on the left. That was the clone on the right. And so they took a somatic cell from the mother, they implanted it into her egg, and then she had a clone that was exactly the same as her. And notice, again, there's some differences. In particular, there's differences in the pattern of the fur color in this case, and that's due to developmental differences. These fur colors, as we mentioned, are due to differences with X inactivation, which X's are turned on and off, and in this case, that's a random thing that happens during development. So even though they're genetically the same, they won't have exactly the same fur pattern. And today, a lot of research in the medical field is looking at this idea of therapeutic cloning and stem cells. And stem cells, the idea is theoretically we could induce those to produce different types of cells in your body. And so if we could obtain some embryonic stem cells through this therapeutic cloning, then we may be able to treat certain diseases. For example, if you had kidney disease and your kidney was failing, rather than go on dialysis and the organ donor list, and certainly most people on organ donor list don't survive to get their organ, and so we don't treat that very well. So what if we could take one of your skin cells, clone it into an embryo, gather a stem cell, and treat that stem cell to grow a new kidney? Now that kidney that was cloned from your cell would have all of your proteins on it, so we wouldn't have to worry about a match or organ rejection, and we could have a kidney then to treat that disease. Now, we know embryonic stem cells can become almost any type of cell. We do have stem cells as adults, but these cells are usually pretty limited. So, for example, we have blood stem cells that produce more blood cells, or bone stem cells that produce more bone cells, but we don't see any cells that produce every type of cell in our body. And so through therapeutic cloning, we want to get a supply of stem cells then to treat various human diseases. And there's lots and lots of research looking into how we can produce these stem cells and how we might treat these stem cells then to get them to divide into kidney cells or blood cells or whatever type of cell we might need. And again, the medical community is so excited about stem cells today because there are so many potential diseases that could be treated by developing new cells that would match the patient's cells, whether they be blood or nerve or heart muscles like we see here. And certainly there's controversy around stem cells today. The main controversy is the source, getting these cells from embryos. But embryos are not being created for this stem cell research. These stem cells that are studied in labs basically come from fertilization clinics. And so when a couple undergoes a fertilization treatment, mom is usually given hormones to release lots of eggs. They're put in a petri dish, take dad's sperm sample and mix them together, and you might get 10 or 20 fertilized eggs. Now normally they're going to implant 3 or 4 into mom's uterus, hoping that at least one sticks, and then they're going to discard the rest of those embryos. And so it's those discarded embryos that are often used for this stem cell research. So these embryos are not being created just for scientific research by any means. Today we might see some changes, or we have seen in the last year, because certain labs have been able to get our human cells to de-differentiate. By treating them chemically under lab conditions, they've gotten somatic cells to unspecialize. And if that's the case, then we may not need therapeutic cloning in the future. We may be able to take a skin cell of yours, get it to unspecialize in a petri dish by treating it chemically, and then taking that stem cell and getting it to specialize into a new kidney or some other new structure. Our last topic then in Chapter 11 has to do with cancer. And we've already talked about cancer a little bit when we were talking about Chapter 8 and cell division. And remember, we said cancer was due to mutations in those checkpoint and control genes that stop your cell cycle. And that each of our cells have anywhere from maybe 3 to 7 checkpoints that our body can stop the growth. And a cancerous cell was a cell that ignored those checkpoints, that our checkpoint system didn't work and it grew out of control. Well, the expression of certain genes are what is going to regulate our checkpoint system. And so the expression of different growth factors or inhibitors are going to turn these checkpoints on and off. And so if there's a problem with these checkpoint genes, then that may lead to cancer. And so gene expression of these different regulators certainly impacts cancer. And there's two main categories of these genes that normally regulate cell growth. One group of these genes are called proto-oncogenes. And proto-oncogenes are genes that code for proteins that act as promoters. That these proteins, when they're present, are going to cause the cell then to move past a checkpoint. So your cell activates these proto-oncogenes when it needs the cell to grow. A mutation in one of these genes causes what's called an oncogene. And onco is just our cancer prefix, right? If you go to the oncologist, you're seeing your cancer specialist. And so if one of these proto-oncogenes, which is the normal functioning gene, gets mutated, we call it an oncogene. And an oncogene then is going to produce this growth factor all the time. And so basically this gene is going to be expressed constantly once it gets mutated, which means that protein is always present and that cell will always skip a checkpoint if that proto-oncogene gets activated all the time. Another class of genes associated with cell growth are tumor suppressor genes. And tumor suppressor genes work just the opposite. For one of these tumor suppressor checkpoints, the cell will normally skip a checkpoint and move past it to the next stage of its life unless an inhibitor is present. And so these tumor suppressor genes produce inhibitory chemicals that can stop cell growth. So if your body doesn't need any new cells, it can turn on one of these tumor suppressor genes and stop cell growth. But a mutation in one of these tumor suppressor genes may result in basically this gene being inactivated, meaning the tumor suppressor gene no longer works and no longer makes the inhibitor. So the end result is, if there's no inhibitor, there's no way for your cell to stop growing. And so your body has no way to stop that growth. And so cancer is due to multiple mutations, and it can be a combination of oncogenes, these growth factors being produced all the time so you skip checkpoints, or mutations in tumor suppressors so your cells cannot be stopped. And so again, cells are going to skip these checkpoints. And cancer is not going to be due to a single mutation. One of these oncogenes or one mutation tumor suppressor will not cause cancer because, again, you have multiple checkpoints. And so even if one of those checkpoint systems in your cell cycle didn't work, your cell could still be regulated or stopped at a different checkpoint. The only time we're going to have a cancerous cell is if all the checkpoint systems are non-functioning in the same cell, and so that is going to require multiple mutations in these tumor suppressors and these proto-oncogenes. And your book looks at the example of developing something like a colorectal cancer. And so we may have a proto-oncogene undergoes a mutation, and so now it turns into an oncogene. It's going to result in skipping a particular checkpoint. And then we may have a tumor suppressor undergo a mutation so that now the inhibitor can no longer work. Okay? And that may result in cells growing faster than normal. And so they're not really cancerous yet. Your body can still stop their growth, but it may start to grow more cells than normal, so that might form a small benign tumor or something like a polyp, for example. Now, as those cells continue to grow, if they have more mutations to skip more checkpoints, so they have more oncogene issues or tumor suppressor issues, then that may lead to a malignant cell, which is a cancerous cell, a cell that ignores all those checkpoints. And of course, every time a cancerous cell divides by mitosis, it makes more cells that ignore all the checkpoints. And so, again, it's this multiple mutation issue. And so we may see a polyp form, and a polyp is not cancerous, but we might call it precancerous. It has the potential to become cancerous cells, because it already may have cells with 1 or 2 mutations in it, and so eventually if they have those last couple of mutations in those cells, they become cancerous, and they're eventually going to grow into a tumor, and if untreated, we know they can certainly be very serious health issues. Now, mutations are rare, but they can happen, and no matter what you do, you have an opportunity or a chance for your cells to have a mutation, but again, it's a pretty low risk. But we know that certain environmental factors may increase your risk of cancer, and so a carcinogen then would be anything in the environment that causes an increase in the risk of cancer. And this would include things like being exposed to X-rays or UV light from the sun, or certainly tobacco smoke has lots of different carcinogens in it. And so carcinogens are going to act to cause mutations in your DNA and increase your chance of getting cancer. And so since cancer is a disease of mutations, anything that causes a mutation may cause cancer. And we certainly know that there's lots of things you can do in your life to reduce your risk of developing cancer. Now, you can't eliminate your risk ever, but you can certainly reduce your cancer risk by avoiding these carcinogens. We know things like high-fat foods may increase the risk of something like colon cancer, for example. So avoiding those types of foods would certainly reduce your risk of cancer. And then lots of medical checkups. We know that today, for many cancers, we're very good at treating them if we get early diagnosis, if we can catch them when the tumor is small and hasn't spread yet. And so certainly that early detection is an absolute key. This chart just shows some data from here in the U.S. and how common different cancers are. And we can see that prostate cancer is the most common cancer, but really there's kind of 4 up there that are all pretty common. Prostate, lung, breast, and colon are all by far the most common types of cancer that we see. And then we see some other types along the list. Leukemias are basically cancers of the blood cells. We have different types of sarcomas, which are basically different types of cancer associated with connective tissues and so on. So lots of different types here, and cancers are typically named after where do we find that cancerous cell, where do we find the tumor in your body. So to wrap things up for this time, we have our 3 big issues associated with differential gene expression. Signal transduction, we know, is due to protein receptors on cells that monitor the outside chemical environment. They bind to proteins bind to these receptors and cause this signal to be passed into the cell, which then is going to influence that cell's gene expression. We know that cloning is a result of gene expression, that specialized cells have certain genes turned on, others turned off. Some organisms, their cells can unspecialize and then respecialize, basically changing their gene expression drastically. Things like plants and a lot of invertebrate animals can do that. But higher animals, like vertebrate animals like ourselves, our cells cannot do that. But we can take advantage of technologies like somatic cell nuclear transfer and clone our cells. And we can either use those for reproductive technologies to clone a whole new organism, or therapeutic technologies to generate stem cells and hopefully try to treat some diseases. And then finally, we know cancer is a result of mutations or problems with gene expression of tumor suppressors in those proto-oncogenes and problems regulating the cell cycle.